Thank you very much. As Gene mentioned, I'm Jerry Sheehan with the California Institute for Telecommunications and Information Technology. I've been asked to speak to you briefly, and I will try to stay on good tone, about the microbiome sequencing work that we've been doing as part of Larry's exploration of himself. And I wanted to talk just briefly about the data that we have. So what Larry did working with the University of California, San Diego Medical School was actually get a sequenced sample that would allow him to look at his microbiome. We did this in collaboration with JCVI. So actually Larry submitted a stool sample, moved to JCVI, and as part of that, what we got back was 230 million short reads, 101 base per reads, and 23 billion DNA bases. Other way to think of this is even in the world of big data, this meant that a 35 gigabyte file was federal expressed to us with ones and zeros that we had to do reconstitution on. So what we were able to do was work with Weizong Li, who had been part of the Gordon and Betty Moore Foundation work that we'd been doing in marine microbial research, the CAMERA project, on a pipeline that he had created for metagenomics. And we were able to take the short reads that we had and move them back to understanding species. And functionally what that meant was about a half year worth of CPU time per sample in order to do this. And when we looked at Larry, and he'll talk to you more this afternoon about what he's seen in the difference in his own data and also some of the comparative analysis that we've done, it ended up that the first sample that he did was at the peak of a flare that he was having in terms of symptoms. Most of his load was actually viral. About 90% of what we saw was viral. Only 2/10 of it was actually human DNA, and the rest was microbial. And so we've got right now Larry from a microbiome gut standpoint through this pipeline at 3 different points in time. We then went back to the NIH Human Microbiome Project, got short reads from them, and ran them through the same pipeline. So we'd have apples to apples comparison, again, about a half year worth of CPU time per individual. And we ended up with 3 distinct phenotypes. So we've got 35 healthy individuals, 6 individuals who have ulcerative colitis, and 5 individuals who have ileal Crohn's at 3 points in time. And that's sort of the data set that we have from a phenotype standpoint. To give you an idea of the taxonomic complexity of what we're looking at, basically what this boils down to is for about 50 individual samples, we have breakdowns to 2,300 species. And what we've been doing at Cal IT2 is working, you can see an image there of the metagenomics research that we've been doing using a 64 million megapixel display, and trying to look at the software tools that would be necessary to do comparative analysis down at the species level in terms of the human gut. So a very quick overview, just to, in a sense, whet your appetite about the complexity of the data that we saw that from a pipeline standpoint is becoming increasingly simple for individuals to do right now. We're certainly doing it at the leading edge in terms of the computational necessities that are there right now. And I'm done before 5.
That's fantastic.
I'm a statistician at Gene's lab, and I'm going to talk to you about the metabolomics data of the cell studies that was published in 2012 on the integrative personal omics profile. So this is a data that was collected on one person. We just consider Mike Snyder here, and it was done over 14 months. It's a pretty complex data set. So they took samples of PBMC, the peripheral blood mononuclear cells, and serum, and used the tissues to extract a variety of omics data, right? So they did genomic study and transcriptomics, proteomics, also autoantibodies, they did glucose tests and things like that. The study covered the period of 14 months, as I said, and included a variety of underlying changes and conditions. 2 periods of infection, the human rhinovirus infection, that's marked in red over here, and then the respiratory infection that's marked in green. Then there were data that was collected when Mike was fasting, and this is the blue days on the diagram. And then there were days when he had certain changes in his lifestyle. So this is the black bar that shows that he increased or decreased the exercise, took some medication, ate more or less sugar and things like that. So it's a pretty complex data with a lot of hidden confounders in them as far as analysis goes. And what we're looking now is metabolomics part of it, and they identified consistently about 6,000 different metabolites in the data. What we did is a little bit of exploratory analysis. It's just sort of at the beginning stage. And when we look at the metabolites, we find 3 distinct clusters. So the 3 diagrams on the bottom show these clusters. And so the horizontal axis is just the time progression, and the vertical axis is the relative expression, and it's relative to the day when the subject was healthy. And as you can see, there is sort of an interesting pattern, especially when you look at the green infection line, just because there is more data collected and it's harder to see at the human rhinovirus part when there's only two or three data points. So what happens is when the infection set up, there is a dip in the metabolite response for the first 2 clusters, and then for the second and third, and then for the second cluster, after the metabolite expression go down, they start to recover. Oops, what was that? And then in the third cluster, the sort of the tendency almost backwards. When the infection set up, the expression ratios goes up, and then as the subject recovers, they start going down. So that's sort of a pattern that we've seen so far. And what was done in the study is they looked at these different omics data and analyzed it separately, and then just sort of tried to integrate results together when they interpret them. But the question is, can you take these different omics data and integrate it so you can increase the power of your analysis and maybe extract more useful information from that? So the one thing you can think of, it's sort of more or less straightforward, is the pathway-centric approach, where you take, for example, metabolomics and the proteomics data, and you map it together to pathways, and then you extract some sort of summary statistics. The difficulty here is you don't have a single data point, right? You have the longitudinal study, so you have to be creative about how you're actually going to do the mapping and extraction, but that can be done, and this is what we did, and I can tell details later if you're interested. And so what we end up at the end is the score for each pathway at each time point that can be thought of as some kind of functional representation of, some representation of the function of that pathway. And so looking at those scores, we, for example, found 6 pathways that show this interesting tendency when the RSV infection starts. At first, the scores are at the baseline level, and then they increase as the infection progresses, and then they decay and go down to the baseline level after the infection starts, or even lower. So this is sort of, as I say, just a very preliminary result. There is a lot of challenges with this data. So there's a lot of data to begin with. There's different types of data. There's a lot of confounders, and by default, it was an observational study. It's not like a hypothesis testing, so it's really hard to build a definitive model here. There is also a lack of reference ranges. In particular, is a populational or personal? So if you look at the diagram like that and you see the pathway score of 5, is it good or bad? Like you have no idea how to work with that. So that would be it, and something to think about.
Perfect timing.
I know.
Thanks, Gene, for inviting me to speak. And in my 2 slides, I want to first talk about some scientific needs that I've recently identified, and then talk to you about a proposed approach for addressing those needs. The first need is to accelerate the discovery of metabolic network defects. There's been a recent new appreciation that metabolism, human metabolism, is a sweet spot in the sense that metabolism seems to underlie many more diseases than was previously thought. Here's a quote from a recent article. It's evident from recent work that many human diseases involve abnormal metabolic states. Now, the metabolomics field is exploding recently and will probably play a large role in elucidating the causes of these metabolic defects. But in general, metabolomics yields very extensive but hard to interpret data sets. And I think we need tools for aiding the elucidation of the metabolic defects that underlie changes in metabolomic profiles. But network defects are extremely hard to identify without precise knowledge of the network. How can you debug a network if you don't understand what the network is? And furthermore, having an accurate model of the network is really critical. Another convergent development is that metabolic pathway bioinformatics and modeling are really maturing. I wonder if you know that E. coli metabolic models are able to predict the lethality of E. coli gene knockouts with 92% accuracy. So metabolic modeling is really advancing. Another need is to fulfill the unrealized promise of personal genomics. The statistical correlations between single genes and diseases simply seem to not be working out as a predictive tool. And I think we need to move beyond the gene prioritization approach. Well, how do we do that? I'm proposing a three-pronged approach, and I'm looking into forming a multidisciplinary center at SRI that will combine work on metabolic databases, metabolic modeling, and metabolic experimentation. The idea is to develop metabolic databases for humans, mice, and for the gut microbiome through an intensive literature-based curation effort where essentially we'll go from metabolic knowledge in this form to metabolic knowledge in this form. And we've developed software tools that allow us to generate from metabolic databases quantitative, steady-state metabolic models. And the idea will be to develop metabolic models for multiple human cell types and multiple mouse cell types. The reason for doing the mouse models and databases, of course, is that a lot of experimentation goes on in mouse, and we really need the same power tools for understanding mouse data as we do for human data. I believe we also need to develop computational methods for personalizing metabolic models. What I mean by that is we need to be able to go from a personal genome sequence to constraints, personal constraints on a metabolic model that allow us to predict personal metabolic fluxes and understand how a person's metabolic fluxes are likely to deviate from the norm. And that will be a way to predict interactions between genes, which are probably the reason that the single gene prioritization approach is not working, that it's really the interaction between genes that yield disease. Finally, we need a wet lab experimentation effort because there are a lot of holes in our metabolic knowledge, so we need to actively seek out through experiment the knowledge that will fill those gaps in our knowledge. One example is pathway holes are reactions in the human metabolic network that have been characterized biochemically, but we don't know what human gene encodes that enzyme. And we can query our existing human psych database to find that there are 227 pathway holes in the human metabolic network. We also need to use experimentation to validate metabolic models and to measure fluxes that will help us constrain metabolic models and make them more accurate. And overall, I see many types of synergy between database curation activities, modeling, and experimentation, in that as we curate new information into databases, that's going to constrain our models. Am I out of time, or where are we on time?
10 seconds.
10 seconds, okay. And then modeling, when a model's predictions are shown to be incorrect through experiments, that tells us new data we need to seek out and new experiments we need to perform to make the model more accurate. So that's all.
Thanks. So what I was asked to talk about is the structure of data in health records. And so there are a series of ontology. I'm not going to use any slides. There are a series of ontologies that have come into common use. The first is the ICD codes, or the International Classification of Diseases, initiated by the World Health Organization decades ago to describe the epidemiology of dead people. It has literally, that was the design principle framework for ICD codes. It has subsequently been tweaked and torqued and twisted to try and describe diseases in the human state, and is now a canonical form for that. There are many limitations of the ability to represent diseases because of its origins in the epidemiology of dead people. Similarly, CPT, or the Common Procedural Terminology, was developed by the AMA to describe procedures, but most importantly, to set up a framework for people who did procedures to bill for more revenue. And so the CPT coding system is, to some degree, a mirror of the problem with our healthcare system today in volume-based reimbursement rather than value-based reimbursement. And it suffers from some of those same limitations in that it was designed primarily for billing. The third one is SNOMED, which stands for the Systematic Nomenclature of Medicine. It is by far the richest. It's translated into 7 languages, used in over 40 countries. And it was developed by pathologists initially to describe pathology specimens, but then has subsequently been generalized to describe clinical conditions, and is the unequivocal champion in terms of its richness of describing the clinical state. And it also has by far the most advanced descriptive logic to be able to operate on through subsumption on it. None of these can support, none of them, each has its own descriptive logic to varying degrees, but none of them can support either negation or disjunction in queries. And so there's another ontology set that allows that, called OWL, and one of my colleagues at Kaiser's developed something called SNOW-OWL, which is a combination of SNOMED and OWL, which allows for the full use of the descriptive logic of SNOMED as well as the disjunction and negation that attends to OWL. A couple of quick distinctions: reference versus interface terminology. So most of these are based upon a reference set of terms. Physicians use different terms around the world, around the country, and so a mapping in the interface terms to the reference terms is available. The third is consumer versus clinical terminology. So consumers developed applications are increasingly trying to develop their own terminology set, and it's one of my missions to converge those two so we can normalize clinician-generated data versus professional-generated data. There is a distinction between structured and unstructured data in health records, and we're required essentially by law in this country and most countries to create structured data elements for diagnoses, procedures, lab orders, and medication orders. And while we have ample opportunity to structure things beyond that, it's completely haphazard. There are some systematic biases of time pressure, but they're in the documentation process and the use of speech recognition for that. So there's a great deal of variation. Then there's sort of a meta structure called the clinical document architecture, which is an XML-based standard that I founded and is used internationally and is required by meaningful use in this country. And a subset of that, proper subset, is called the continuity of care document, which is a list of just the discrete elements. Within that is a mechanism to retain the link between a summary level data element and the source document. So if you've got an allergy and it came out of a note, there is a structure within that construct to be able to retain those linkages. It's only deployed as of today for advanced directives for care around death, but it is available to others, and so it's not well implemented at all. Finally, there's several new initiatives, one from HL7, which is the parent of the clinical document architecture, called FHIR, F-H-I-R. There's another one that's underway internationally to try and normalize different data sets from multiple different countries, called CIMI, C-I-M-I. Thank you.
Okay, thank you. It's a pleasure to be here. If I can only see this. Okay, well, I'm going to pan out a little bit and speak to the issue of context of what we're talking about in this meeting, and I think this, what I'm going to address, will sort of addresses both this and this next quantified ecosystem that you have on there, but there's, I think, an important interface here. And I like this, this quote from Francis Collins, that genes are the bullets, but it's really the environment that pulls the trigger. And I came across this construct actually developed by Christopher Paul Wild, who's a molecular epidemiologist in England, about the exposome. And it's really everything that we are surrounded by that ends up triggering the various sorts of things that might happen related to genetic predispositions and other phenomena. And it's really the accumulated exposure that people have. So it's essentially the area under the curve of a lifetime's worth of bad eating, insufficient activity, a variety of things. And so the behavioral interface, and you saw in Dr. Snyder's discussion of the issue of lifestyle. So lifestyle is actually pretty important when we begin to think about this. And if we are talking about developing methods to better understand and or intervene in this area, we've got to find ways to wrap our mind around what the exposome is. And we're doing a fair amount of research at UC San Diego in this. One really exciting project is this project called CitySense, where we've developed a portable sensor board that captures carbon monoxide, ozone, NO2, temperature, humidity, and whatnot, and it radios that via Bluetooth to a mobile phone, can give people feedback right away, instantly, of the air quality that they're exposed to. This can also go up into the cloud and be mapped with other users as well. And it can be shared, and so there's a social dimension to this as well. And this actually is a one-of-a-kind project. We've looked around the world, and no one is doing this in quite the way we're doing this in terms of real time. And we see this, there's a lot of excitement about this in terms of what people might understand about what they're exposed to. Another project we're working on is looking at this compelling problem of obesity. And so we've got this mashup of an intervention of Facebook and smart apps and wearable sensors and whatnot, and we're focusing on this issue of weight gain in young adults. And again, the behaviors that are associated with this are extremely important, and we're learning a lot about what it is that undergirds this. So again, this is how we're looking at the context of all of what's being discussed at this meeting, and I think in the overall framework of DELSA. And based upon this work, we have just begun a new project, Jerry and Larry Smarr, and I, with support of the Robert Johnson Foundation. It's a planning grant right now to begin to explore how we, as researchers, get closer to the forms of data that are being captured and collected by people on their own. 20% of the people in this country now are using some sort of device to capture some form of health-related information for themselves. That came from a recent Pew Foundation report. So we want to explore whether, and I would say a lot of those data, you heard John talk about health-related data, a lot of the data that are going to be important to health are happening now outside of the context of traditional healthcare environments. And so these vast amounts of data that are being built up need to be opened up, we think, to researchers. And so we're going to be digging in on this, trying to get some sense of what the mindset is within the private sector and these data-rich companies, everything from Facebook and Twitter and Google all the way down to Fitbit and Nike Plus and whatnot, and see if we can develop methods of opening those data up via data use agreements, potentially technical assistance, finding ways to sort of break down this barrier that might exist. And then one final thing we're looking at is this issue of potentially developing a data commons where people could donate their data. We've already got Larry doing this, we've got people like Dr. Snyder doing this. More and more people do this, then the insights that we can gain from this could be enormous. But the devil's going to be in the details. How do we actually get from here to there? So the Robert Wood Johnson Foundation, the leading foundation in the country related to public health, has taken the lead here and is very interested in seeing this happen. So we're open for suggestions and ideas about how we might do this in the most productive way. We're going to be unveiling our website on this in the next week or so, and Judith Gregory, I see, is there. She's going to be helping us as well from UC Irvine. This is a Cal IT San Diego Irvine project, and so please feel free to let us know about how you think we might succeed with this process more appropriate, more strongly and better. Okay? Done.
So this might be a slightly different type of talk. I just had the pleasure of meeting Gene over the last couple of months, and he asked me to give an overview of the type of work we're doing. So the type of modeling that I do is really systems engineering, by which I mean operations research. So that spans everything from process optimization to simulation and statistical modeling. And we've been building an institute at Northeastern University, as well as building a formal and informal network of systems engineering centers across the country to try to drive this into healthcare. And really, the focus is really more on optimizing the delivery system of healthcare. So the healthcare system, or the WellCare system, when we're focused more on population health and keeping people out of the healthcare system. So you can see our mission there, and it's a typical trilogy. I mean, we have mechanisms and centers focused on pure research, methods development to solve problems that we don't know how to solve, as well as a fair amount of funding from the Innovation Center at CMS to really run demonstration projects about the potential impact of driving systems engineering, sometimes called industrial engineering or operations research, into healthcare to optimize these types of systems. And all of that is really based, and then I'll tell you a little bit more about, I think, why I'm particularly interested in this community related to some of the research that we're doing, because we're doing all of our work based on a broad network of scientists and delivery systems to try to elicit from them their real problems and work with them sort of in their systems to optimize them. So this is just a quick look at, I've sort of snuck 3 slides into 2, as I've noticed some other folks have done. The initial, we have a National Science Foundation Center called CHOT, which is really a network of researchers at now 6 universities and healthcare systems all over the country. So much like this community, how do we keep them together? How do we form ties and binds formally and informally for knowledge exchange? So I'm very interested in that particular type of problem. Here are just 4 types of problems we're working on. Everything from kind of the macro level optimization of a large delivery system. I mean, it could be Kaiser across the West Coast. It could be the VA healthcare system in the New England region. And how do you model that mapped against where people live and their demands for different types of care in the upper left-hand quadrant here, and design a system that has enough capacity to provide the right care or well care, either at home or using remote technology, telehealth, or when they need to come in, they need to come in at minimal cost. And of course, what's fascinating about our particular healthcare system in the U.S. is typically you have trade-off frontiers where you have to play one criteria against another. But typically, we're able to simultaneously significantly improve cost, access, and care with no particular trade-off against the other. Down to in the right-hand upper quadrant, really microprocess optimization. How do we schedule a whole clinic or system of clinics around family medicine and primary care and preventative care to maximize continuity? So this type of problem is so that a patient can see the individual that they would like to see, that they're familiar with, when they would like to see that individual, because there's a lot of literature that doing that leads to better trusting relationships and better preventative and compliance measures in terms of all the behavioral things that drive the need for episodic care. And as well as there's a lot of, when you look at the United States as a system or a system of systems, it's an economic game theoretic type of problem where we have a lot of cooperation but a lot of competition. And how do you use incentive systems and design optimal economic incentive systems to maximize the whole puppy as a whole? Now, we're particularly interested in this lower left-hand quadrant. Like waiting for the beeper to go off here. But we're trying to evolve a broad network of folks doing this type of work, and we're also trying to study the topology of those networks using like social network mapping types of tools. And I'd be particularly interested in sort of helping us think here about how we evolve over time, formally and informally, what types of topologies drive and correlate most to the richness and quality of this network, knowledge exchange, advance of science, et cetera. So that's just a snapshot of the type of work we're doing. Thank you.
Right? So please,
So my
introduce
question,
yourself.
sure, Eric
shortly.
von Schweber from Surveyor Health. My question is for Kevin Patrick, and I was wondering, Kevin, what sort of incentives are you contemplating? I know we're involved with companies like First Data Bank, et cetera, and there is, because of no financial motivation, everyone is maintaining stovepipes, as opposed to saying, if we could share in some revenue, we'll share some data. So I was wondering what sort of incentives or motivations.
And this would be motivations for the Robert Wood Johnson project that I mentioned?
Yes.
Yeah, well, that's a very good question, and I think that's what we want to, what we would like to get suggestions for in terms of what it is that might work. I mean, people will donate things out of altruistic reasons. You know, we think some people will do this, but whether companies will do that is a whole other matter. And so I think we'll be testing the waters of the various cultures. You know, could there potentially be some added value that could be provided back to that company in some way based upon participation in a commons? Again, right now this is a tabula rasa. We are very, very open to suggestions and open. We're comprising a board of directors. We've got people like our board of advisors. Tim O'Reilly is signed on now, and so he's got some interest. Linda Avey, who is one of the co-founders of 23andMe. We've got Mary Wofford on. So we've got some. We think we've got some pretty. Gary Wolf from Quantified Self. A lot of this comes out from the Quantified Self movement. I think we've got some good thinkers who will be giving us some advice, but this is what this 6-month planning grant is about, and it's very, very open to ideas and thoughts. And then if we're successful with this, what Robert Wood Johnson Foundation may do is then move forward with some sort of program that could well be mini grants. They could support some work within academic settings or others to dilate and build this out. But again, happy to go offline, and please populate. Again, when the website goes up, there's going to be places to enter in suggestions and comments about what we might.
So I just wanted to make one comment. I think there might be an opportunity in the last few years. So if you think about, since you're sitting next to John, and John was talking about, well, we need to go beyond current vocabularies and lexicon to true ontologies, SNOMED plus OWL. There's been a whole front that's been afoot in the last few years called linked data and linked open data, supported by the formal semantics from W3C, like OWL and RDF. And so far, it's all been about open and free data. And there have been some attempts in Europe to create some, let's say, for-profit, not open data, but private data that has paywalls associated with it, either on a macroscopic scale or granular per data element that are shared and retained. And it might be that by taking John's direction of trying to move medical vocabularies into a formal semantic space and coupling that with not just open linked data, but open and mixed private data, there might be an incentive to say, hey, you can get this free, it's easily accessible using commercial and free APIs, sort of like an on-ramp into then the data you pay for, so that there's a motivation for large enterprises to participate.
Yeah, let me respond real quickly to Eric's comments. I agree with everything you said, and I think we need to do all of that. The challenge in my mind is results from something that's been predicted for years but was published in Science in February of this year, where you can take a de-identified genome and re-identify individuals and families from it. And I wrote a chapter in a book on that very issue, submitted the galleys the day before that article came out, positing that the value of the kind of linked data that we have and what we're all trying to achieve through the linkage of the data is very much depreciated if there isn't a multi-omic signature within that data set because of all these interactions. Once you do that, and once you start aggregating data from multiple sources, it's virtually impossible to prevent re-identification. So from a public policy standpoint, in order for us to be able to incentivize the creation of the donation and the pooled data across all of the aspects of human data collection, we need to have an entirely different zeitgeist and policy around the fact that security is necessary but insufficient, and that transparency into who has my data, what they're doing with it, and severe sanctions for abuse that has not been authorized is, I think, going to have to precede or at least parallel the creation of what you just described. So I'm all in favor of it, but I'm in favor of a much more candid discussion about the fact that prevention of de-identification, prevention of re-identification is not at all addressed in a sufficient way in current legislation.
Bill Hancock, Northeastern University. I wanted to make a comment which is either obvious or controversial, one of the two. With all the complexity of the genome or exposome or whatever, I would argue it's only significant if it changes the isoform population of the some 5 million protein parts, and that should link into the metabolome. So with all this complexity, I would say the protein isoform is the central thing, and if it changes, the metabolome is going to change. If it doesn't change, it's just complexity.
panel or from the audience wants to respond. So if nobody, it looks like it's not controversial. It's kind of statement then, right, Bill? No, we do have.
Was there a question in that?
Yeah, we do have microbiologists here. So if you can maybe reformulate as a question.
So we have all of this complexity. All I'm trying to say is that if it changes alternative splicing or changes phosphorylation or glycosylation, amino acid polymorphism, actually not at the transcript level, but actually getting to the protein level, it has biological significance. If it doesn't have biological significance. Maybe that's a little more controversial.
So you're talking about the epiproteome as opposed to the epigenome. Phosphorylation, glycosylation of proteins and so forth.
Well, it's just that
Analogous to methylation of DNA. I mean, I'm just trying to draw some sort
Okay,
of parallel.
I think I got the question. So I think some of us would agree with you that, you know, if it's not translated on protein metabolic level, it's not biologically significant. And I'm sure that some would disagree with you because they think that regulation happens on that level as well. So with maybe without very clear consequences for our current understanding of molecular mechanisms. So I would say that it's really complicated, loaded question, which I'm not sure anybody can resolve right now.
But I would argue from a biological context, since pretty much everything is mediated by a specific isoform of a protein, if the isoform population doesn't change, you're not going to get a biological effect.
I would agree with you, but again, I'm sure that part of our community would not. Okay, so Eric, please.
Hi, so my name's Eric Schultes, and I'm coming from Leiden University Medical Center. And I will comment on Peter Karp's talk because this was extremely apropos to our own research. But then the first conversation that was here with the idea about trying to incentivize knowledge sharing, yet at the same time somehow preserving the profit-making potential of proprietary data. We also at LUMC are trying to come up with a solution for this problem. We call it nano-publications, and I'll be talking about this tomorrow, but it's essentially trying to augment linked data with provenance, and then attaching to it licenses that would restrict who can actually see what they're looking at. And the idea would be to share information as much and as open as possible, but then if you really want to know the details of some semantic reasoning on the network, you can go back to the provenance and then pay, say, Elsevier or Nature Publishing Group for the specific article so you can get the details on it. And yeah, I'll talk about that tomorrow.
Is there atomic level provenance or just aggregate level provenance?
So it can be, so it's you know, the nanopublication model is basically just a schema that sits on top of semantic technology. So it's really extensible and open. The provenance can be anything you want it to be. The more clever you are in your provenance, maybe the more clever, or the more often you would get cited, and there'd be bonuses that way too.
You wanted to comment on Peter Karp, right?
And now I will comment on Peter Karp's talk because this was like exactly what we're doing. Now we have a paper in review at Nature Genetics where we text mined PubMed, and we created a list of gene disease associations from that. And it depends on how stringent you want to apply the statistics, but we were saying in this paper that we have identified maybe 1.5 million gene disease associations that are probably likely or meaningful in some way. When we submitted this, the reviewers came back pretty critical, saying, I can only test 1 or 2 gene disease associations in my lab in the next 10 years. So I don't need 1.5 million. You have to prioritize these. You have to tell me which gene disease association I'm going to do next. We're lucky to have a big friend in the editor at Nature Genetics who understood that we're not prioritizing gene disease associations, but we're doing something different. And we're using the language now of a literature-wide association study in contrast to the genome-wide association studies. So the idea is just to say, hey, these are all the associations that pass some threshold. We don't know what to do with them. We don't know their significance, but they're there. And then what we're offering now is saying, okay, if we have LWAS and GWAS and we look at their intersection, maybe the things that are in a consensus between those two groups become what we test.
You wanted to ask some question? I'd like actually everybody to be concise with their time because we want other people to have chance to ask the question. If you give presentation tomorrow, don't tell it today. So if you have specific question to Peter, that would be great.
No specific question to Peter, just a comment on something that looked very similar to what we're doing right now.
Thank you, Eric.
Yep.
Viral.
Viral Özdemir, McGill University. I just want to thank all the lightning talk speakers, and a comment I had that seems to be emerging for all the talks, which is the importance of the commons in 21st century science. And the new way of doing science, I think there were really fascinating sort of talks, including on exposome and new ways of working. But really, the name of the game is commons. So I tend to think that DALSA Global is really contributing as a commons maker, if you will. And we really need that. We need that kind of an honest broker that can build the commons because it can benefit many. And a related comment is that the name of the game is not only commons and building the commons, but softer things that are actually quite powerful. In other words, things like competition and cooperation. In the old way of doing science, it was either that you would compete with certain people and you cooperate with others. But it's now more fluid and more dynamic. There are things like pre-competitive collaboration. But I will argue today that we also need new concepts such as post-competitive collaboration. In other words, can you collaborate after you competed? So we need to understand these things. And again, DALSA Global is not only a commons maker, but is really a forum where you can study these kind of emergent phenomena. Thank you.
Thank you, Viral. Banu, please.
This is Banu Rekpalli from Joint Institute for Computational Sciences, University of Tennessee, Oak Ridge National Laboratory. I have a question for Peter and Larissa. I've been hearing about this big data size is really big. So how do you anticipate, is the growth will be similar to genomic data growth? And how big is this data? And what are the bottlenecks in computational methods to solve or analyze this data?
My answer is probably simpler, so maybe I'll go first, which is for the databases that I'm anticipating, they're actually relatively small. For example, I've worked on an E. coli metabolic database for many years, and the E. coli metabolic network is not that big. It's on the order of 1,000 reactions and 1,000 metabolites and 4,000 genes. It's more, what we've developed for E. coli is a very rich database with over 300 different fields. So it's a much higher ratio of data fields to data instances than is typical in other areas. And I'm really anticipating extending the approaches we've taken for E. coli to humans. Now, it's true there are many human cell types. The human genome's bigger, but maybe it's a factor of 10 or a factor of 20 larger. I'm focusing more on building rich databases that can be used to interpret the high throughput metabolomics data that people like you are developing.
So the data set's getting very large. I think Larissa Smarr is another few gigabytes, right? The one that you have to shoot back and forth.
Just the metagenomic sample, the single sample was 32 gigabytes. And I think the other thing that's important from our perspective is that really the challenge here is to begin to think about systems biology and an integrated way of looking across all of these components. And that's a much bigger data challenge than any particular sub-area that we might have. And that is, I think, the meta challenge moving forward.
Right, and so integration where it's, the data size become again very important. When you have thousands of data, like a lot of data analysis methods that are out there, they're not really meant to deal with the, thousands and thousands of data points that you have, especially if you deal with something like longitudinal analysis, like how you go about that. There's a lot of tweaking, like even, like as simple as clustering, right? It cannot just go and crunch through 100,000 proteins easily and give you some meaningful results. Because what you want at the end is just, 4 or 5 definitive patterns that you can interpret. You don't want 90,000 of them. And that's, that's where the bottleneck is as well.
Todd, please.
Yeah, Todd Smith, PerkinElmer. A general question is when we're thinking about linking an observation, metabolite, all the way down to the genome, and this also is, how we contrast to like astronomy, is are we using the right kinds of telescopes, right? So that is a problem in biology, and to your question, is you have multiple redundant pathways that can produce a common metabolite. We can have multiple genotypes express a common phenotype, and sorting that out becomes very difficult. And Peter, in E. coli, how many times do you see multiple redundant pathways in that list of 1,000 producing a single measurement? So I think, as we go forward, we need to think also about these measurement systems in some interesting and detailed ways. Hypertension as a classic example in medicine, yet people can get just on a therapeutic odyssey trying to dial in on that. So that's sort of a statement and a question. Any thoughts on your telescopes?
Well, in general, I see this as one of the main roles of metabolic databases, is to connect metabolites with the genome, right? Yeah. You need to know which genes code for which enzymes that catalyze which reactions that generate which metabolites. And if you don't have that connection, you can't go from changes in metabolite patterns to the underlying causes at the proteome level or at the genome level. So that's why I see metabolic databases as essential.
But the shunts kind of, I measure a metabolite, but I could have two pathways.
It's true. There's ambiguity. That's why it's non-trivial to debug these networks.
One thing I'll add is the so what question here, I think, is very proximate. And the so what question is when we treat hypertensive patients and we encounter this kind of redundancy and complexity of the physiologic pathways, I think as we begin to unravel this mystery, we're going to see a lot more what I call hyperpolypharmacies, where to address the issues of the redundant pathways for creating a certain state of health or a certain state of disease, understanding those redundancies and complexities may allow us to have deliberate, very focused approaches that are both personalized to the individual as well as respective of the redundancy of some of those pathways. It's not something we do well today.
So I think, Jim, you were not that lucky. So I have a question for you, if that's okay. So one of the, most important lessons learned during your analysis of other communities, other complicated systems, whether we talk about DELS as a community or we talk about our organisms as, complicated systems, can you tell us something on that one?
Well, we're early in still, but this is the classic problem, which is the translation of knowledge across communities for mutual or different purposes. So the knowledge exchange networks we're looking at, you see a lot of clustering, and grouped in different ways. Which when you look at it is obvious, sort of retrospectively. So what's interesting to me is once we understand sort of the deficiencies in a knowledge exchange network, how do you intervene and act on that network to maximize, the purpose of it? And that's what we're really interested in studying. I mean, I'm just learning about your community here, and apparently you're kind of going global. Right, so I mean, it's natural to think that there's probably clustering by continent, et cetera, and then so what actions can you take on that network to maximize it?
So I'd like to ask a question of both the panel and the audience, but the extent to which you can exploit any data set depends to a great degree on to what extent you understand the constraints of how the data was collected, how the data is represented, how the context is preserved, how the linkages are preserved within that data set. So my question is, does anybody have a good systematic model for how to represent data set constraints based upon all of those parameters? Because I think that's going to be an increasingly valuable asset that attaches to each of the data subsets that we operate on, so that as we do cross-data set queries, we have some understanding at least of the primary constraints of the individual data sets, let alone the interactions of those constraints across data sets. Does anybody have a model towards solving that issue?
Larry Lanham, CNRI. I don't have a model, I have an organization, however. One of the hats I'll be wearing here is Research Data Alliance, which just came into existence, NSF, EC, Australia funded, expanding, and that's exactly one of the problems they'll be looking at. Both citing the data, that's a group called DataCite has started working on that, but then explaining the data, metadata, identifiers, et cetera, across scientific data, if possible. The organization's just coming up, trying to figure out how to organize itself. What it's worked, her first few working groups have come into existence and so on. So I encourage interaction with that group.
So as someone who loves taxonomies, have you come up with a name yet for the problem I described?
No, just infrastructure, you know,
okay.
E-infrastructure, cyber infrastructure.
I think that would be helpful to aggregate people's focus on it.
I would think the name is metadata, and it occurs to me that actually that makes the de-identification problem even harder, right? Because if you just have, it's easy enough to identify somebody from their genome data alone, but if you have the extensive metadata that you need to put that genome data or transcriptome data or whatever in context, that makes it even easier to identify the person, presumably.
So one of the things I would argue a little bit about is that I think metadata is a proper subset of the problem I'm trying to describe, because I think there's metadata about how the data was acquired that is not necessarily capturable in metadata itself.
Yeah, you have to understand how to bootstrap.
Meta models?
You have to understand, we need use cases and how to bootstrap up from, you know, here's some data, what the hell does it mean?
Yeah, so this is the key issue. I'd say the key bottleneck in nanopublishing is what we call it data modeling. And so we're a nanopublication we think of as an assertion. So, you know, what is the data? What is the observation you want to talk about? But then there's the provenance. So who are you, and what was the experiment or the context in which that data came from? And yeah, we see this as the bottleneck. But we can offer some examples of data models that we've created where we are trying to show interoperability between ENCODE data and Phantom 5 data, for example, or other genomic or pharma-related projects.